Thank you very much, Dr. Brower. And it's a real pleasure to be with you here today at the 2021 Newborn Screening Summit. Again, my name is Dr. Timothy Yu. I'm a geneticist and neurologist at Boston Children's Hospital. And the title of my talk is going to be Actionable Genetic Findings in an Era of Gene-Targeted Therapies. The goal of my talk is to discuss recent advances in therapeutic development that are transforming our ability to treat even the rarest genetic conditions. And I will present a few case studies that illustrate this and invite you to think about and then engage in a discussion in the Q&A afterwards about how these new capabilities might alter the way that we think about what we consider a quote unquote actionable genetic finding, and how they also might invite us to consider possible impacts on how we assess risks and benefits in early detection and early screening and newborn sequencing strategies. By way of background, I am a neurologist. I run a research laboratory that does both basic and translational work at Boston Children's Hospital. Here is a snapshot of my group in the pre-COVID days, and here are a few of the topics that we have worked on, including basic gene discovery to understand mechanisms of human brain development, diseased gene discovery in the realm of autism genetics, clinical translational genomics, developing methods for genome analysis and interpretation, and increasingly in the last several years, we've focused our efforts on translational genomic medicine. And the general driving theme of our work has been, how do we actually take the fruits of genomic medicine, the fruits of the genome sequencing revolution, and apply these in ways to bridge the gap towards patient care. And in our thinking about this problem, we note that have been extremely lucky to live through a period when genome sequencing has enabled the basic scientific discovery of a host of new genes responsible for human disease. We've also been fortunate to live through a period where the gradual clinical adoption of genome sequencing is revolutionizing the diagnosis of patients with these conditions. And we are now increasingly confronting that remaining gap, which is that developing treatments for these conditions still takes a long time. To illustrate this, drug development historically has some really sobering statistics. It's been a slow and painstaking process where one identifies a condition that one is hoping to treat, designs a therapeutic approach, demonstrates an experimental proof of concept, then has to navigate multiple steps to manufacture a clinical grade drug, to have it tested and formulated, safety studies in animals, phase 1, 2, 3 trials, and hopefully approval at the end of the process. And all of this work has historically taken great expense with really staggering costs of average costs per new drug approval of over $2 billion, extended times of over a decade from target discovery to approval, and a really disconcertingly high failure rate. These are statistics, however, which are from 2014. And the good news I have to share is that in 2021, this is beginning to change. Now, that change is being driven by many different factors, one of which is the fact that genetically targeted modalities are showing fantastic promise in accelerating this process. By gene-targeted or genetically targeted modalities, what am I referring to? Well, I'm referring to a host of different drug development technologies and platforms, ranging from gene therapy, which you may have read about often in the news, as a way to deliver genetic cargoes into the body of patients with genetic mutations via a virus to replace something that might be missing. There's also mRNA therapies, which have been a subject of a lot of attention in the last year, especially because of the current COVID pandemic. Antisense oligonucleotides are the subject that I'm going to be focusing on. Also, other things called siRNAs for silencing certain genes, or CRISPR genome editing, where we actually go in and change the fundamental DNA code of an individual to help try and ameliorate some clinical condition. These are all examples of novel genetically targeted modalities that are promising to change the way that this practice is delivered. As I mentioned, by way of my case examples, I'm going to be focusing on antisense oligonucleotides, one of the most rapidly maturing of these platforms, to illustrate a few points. By way of background, antisense oligonucleotides are a mouthful, but they're often abbreviated ASOs. What these are short snippets of synthetic DNA or RNA that are typically around 15 or 20 letters long. What's remarkable about these kinds of drugs is that they're easy to manufacture. They can be manufactured using machines that can spit them out in less than a day. And you can design them to be manufactured with a certain order of letters that allows them to home in on specific genomic targets using traditional high school biology Watson-Crick base pairing. The patterns of A's, C's, G's, and T's dictate where these drugs go. Along the way, this fundamental targeting technology has been enhanced with chemical modifications that give these drugs good biodistribution properties. They're well absorbed. They've been modified with modified backbones and slightly modified nucleotide bases to have good absorption characteristics, broad distribution, good stability, and good safety indices for human clinical use. And these types of ASOs have multiple applications for addressing root causes of genetic diseases. They can be used, for instance, to boost gene expression in some instances by changing the way that certain genes are spliced or assembled together. They can also be used to shut down genes by, for instance, reducing the amount of a toxic gene. These ASOs can do this by homing in on certain specific gene targets in such a way that it engages parts of the cell that now recognize that gene for destruction, causing that gene to be so-called knocked down. This is an example of how you can deliver one of these ASOs. For delivery to the central nervous system, for instance, neurologic disorders, a spinal tap, a small needle, can be advanced into the lower spine of a patient. Here in the lower back, a needle has been inserted and then into the space of the central cerebral spinal fluid, which is the fluid that bathes the brain and spinal cord. And injection of an ASO drug there will lead to spread of that drug up the spinal cord, up into the thorax, into the neck, and eventually up into the brain as well, where here we've got a graph showing that these are levels of the detectable antisense oligonucleotide drug seen in these various tissues, in the low spine, the mid spine, the high spine, and then the cerebellum, pons, and cortex, 3 parts of the brain from base to top. And once it's distributed along that axis, within the cells themselves, it's broadly taken up throughout cells of the nervous system. These are brain cells looked at under high power microscopy, showing that both neurons, which do the computing of the brain, as well as supporting cells, the glia of the brain, are avid. They take up this drug quite effectively. And what you're looking at are brown staining patterns representing an antibody that is designed to recognize an antisense oligonucleotide. And this brown staining is seen in all of the neurons, the brain cells in this field, in the low spine, the mid spine, the high spine, the brainstem, and in this case, motor cortex. One of the features of ASOs that makes them remarkable is that they are easily customizable for different genetic targets. And I alluded to this by describing how they are short sequences that can bind by via Watson-Crick base pairing. By example, Here is an example of an ASO sequence which targets a gene called SMN1, which is the cause of a severe infantile genetic condition called spinal muscular atrophy. Below it in blue is a different sequence. It's exactly the same length. It's a different sequence, but this drug, by virtue of its different sequence, targets a different gene called CLN7. CLN7 is a disease, is the cause of a disease that I'll tell you in much more detail about, a neurodegenerative disease that unfortunately affects children. These two drugs use the same fundamental technology, the same building blocks, but they have a different sequence and therefore a different utility, a different clinical utility. To illustrate how this works, the best described example of an antisense oligonucleotide drug is one that was developed recently for spinal muscular atrophy, or SMA. This is a disorder in which babies are born weak and hypotonic with low muscle tone. And unfortunately, untreated, it progresses to death from respiratory failure. This respiratory failure and this muscle tone is caused by loss of a gene called SMN1, which is a survival factor for motor neurons that control our muscles. And these babies who are born with this, as I mentioned, have respiratory failure by age 2. Now, luckily, our genomes encode a backup copy of SMN1. That backup copy is called, creatively, SMN2. It's 99% identical to SMN1, but evolutionarily, it's been rendered inactive by a C to T mutation, a single letter change in the seventh exon, the seventh instruction coding cassette of the SMN2 gene. And this single letter change, a single C to a T, causes the SMN2 gene product to be assembled improperly, such that the normal sequence of instruction sets that go from 1 through 8 are misassembled, such that it's assembled in a pattern of 1, 2, 3, 4, 5, 6. It skips 7 and then goes straight to 8. And that pattern unfortunately results in a non-functional SMN2 protein, meaning that this backup copy doesn't do its job. Nusinersen, or Spinraza, is an ASO drug that restores SMN2 splicing. It's a sequence here, which is 18 nucleotides letters long, and administered to babies given by spinal tap 3 times per year. It homes in on this spot where this exon 7, has been altered. And by binding to that spot, it can have the impact of changing the splicing pattern, the assembly pattern of the SMN2 gene, such that this antisense oligonucleotide in green here now allows exon 7 to be properly included in the instruction cassette, makes functional SMN protein. And in a really, truly remarkable set of clinical trials that began to be published in 2013 and extending into 2017 and 2018, clinical administration of this drug to patients with SMA showed remarkable gains in their ability to gain strength and their ability to support their weight and to support their own breathing and to walk and to run. It's been really a game changer for that population. SMA was the first example of an ASO drug that was approved by the FDA, but there are now a wide variety of drugs that have been FDA approved or are looking promising in various stages of clinical development. And this slide just shows an example of the wave of drugs based off of this technology that are being developed for conditions like ALS and Huntington's and early infantile epilepsies like Dravet syndrome, as well as FDA-approved drugs for SMA and these conditions here, TTR amyloidosis and familial hyperchylomicronemia syndrome. And this approach of using ASO drugs is even promising to open up avenues for treating really unusual situations of ultra-rare conditions that previously were considered too small to merit sustainable commercial development. In fact, in 2017, we we encountered a case of using an antisense oligonucleotide drug for even an individual patient, which I'll describe to you here. Back in 2017, my team happened by luck to come across this post on Facebook, a post on social media, describing a family that lived in Colorado whose 6-year-old daughter had just been diagnosed with a fatal neurodegenerative condition called Batten disease. And their physicians were recommending whole genome sequencing to help understand this diagnosis. And we're looking for help for someone who could help them perform that sequencing. We investigated this case and we learned about this child whose name was Mila. And she was at that point a 6-year-old girl who had suffered vision loss and neurologic decline over several years. And she had been diagnosed with a condition that was unfortunately inexorably progressive and predicted to be fatal within a short number of years. However, her diagnosis was incomplete, and they needed to complete this using whole genome sequencing, which we then decided to undertake. And we were able to perform whole genome sequencing on this child and come up with a precise genomic diagnosis that not only solved her case, but also created a very unusual opportunity. The normal CLN7 gene is shown here. It's got 13 instruction coding cassettes. And what we found in Mila's case was that she had a misplaced segment of genetic code causing missplicing in between cassette 6 and cassette 7. And this finding, which wouldn't have been possible without performing whole genome sequencing, actually resulted in abnormal assembly of her CLN7 gene, such that it now was truncated. It now no longer bridged exon 6 and 7, but instead spliced into this abnormal sequence, which was activated by this genetic mutation. And what we realized was that this actually created an opportunity for us to use the antisense oligonucleotide technology I just mentioned to create a drug to fix it. This case, which has been now received a lot of media attention and widely publicized, led to us being able to go from initial patient referral in January 2017 to developing a truly individualized medicine for her in just 1 year's time. And this effort began with identifying the mutation, realizing how it worked, conceiving of a hypothesis for how we could fix it, designing a drug in our laboratory, testing it and proving it was working in our laboratory, manufacturing it, and doing safety studies in animals and treating our patient by the end of January of the following year. And that effort leveraged a number of exciting properties of ASOs as a therapeutic platform, the fact that they're rapidly customizable, the fact that they're actually simple and relatively inexpensive to manufacture compared to many other types of drugs, the fact that they're easy to deliver, and that they have a growing safety and efficacy record, making these, as I mentioned, ideal for tackling drugs previously thought to be too rare for commercial investment. This effort, which was led by members in my laboratory, including April Hu, Aubrey Susi, Jay Vase, as well as Jinkook Kim, who's not pictured, relied on our ability to computationally design these drugs and screen them in our patient's own cultured cells using a skin biopsy from Mila, and using these as a test bed to look through the drugs we had designed and select those that had promising efficacy. And all in all led us to begin treating our patient with very gratifying clinical results of improvements in seizures that were quite dramatic over even the first 6 months of treatment. We saw her seizures plummet from some 15 to 30 down to 0 to 10. And this work was published in the New England Journal in 2019. Now, without going into details, this approach has been the beginning of a wave of antisense oligonucleotides that have been developed for individual patients, including the antisense oligonucleotide drug jacefusin, which was developed jointly by Project ALS, Ionis, and Neil Schneider on behalf of a 20-year-old patient with a severe and genetically aggressive form of ALS. And it's also been followed by development of another patient-customized antisense oligonucleotide drug. This one published in preprint form just earlier this year by Dr. Bob Brown and Jonathan Watts of UMass Medical School, in which they also developed an antisense oligonucleotide for another patient with a genetic form of ALS due to mutations in the gene C9orf72. Here, we ourselves have pursued this line of investigation for a patient that I'm introducing to you in this slide. This is a 4-year-old girl with Ataxia Telangiectasia. Her name is Ipek, and I'm sharing her name as well as Mila's and her photograph with her family's permission. She has a condition called Ataxia Telangiectasia, or AT. It's a genetic disease. It causes cerebellar neurodegeneration, which manifests as progressive motor incoordination and impairments in her ability to walk, eat, swallow, read, and speak. And here she is at age 4. Typically, this disease is actually diagnosed around this time at age 4. And unfortunately, the prognosis for this child is that she will be wheelchair bound by age 10. And ultimately, it will be expected to be fatal in her 2nd, 3rd, or 4th decades of life. This is an exceedingly rare condition with 40 to 100 US infants diagnosed per year. And there are unfortunately no effective treatments for it. However, what's different about Ipek is that in January of 2020, at age 2, we were able to initiate an investigational antisense oligonucleotide treatment for her, giving her certainly no guarantees, but a chance to be different from the typical prognosis of this condition. And this investigational treatment was designed by our laboratory to correct an underlying genetic mutation, which I'll describe to you. And it used exactly the same design and testing strategy that we employed for Nusinersen. This effort, again, which has been ongoing for the last year, is in its early stages. We can't claim by any means success or that we've prevented disease in her, but it has been well tolerated to date. And I'd like to highlight, for purposes of this summit, how did we actually get to the point of treating her at age 2? If AT, as I mentioned, is usually diagnosed at age 4, how did we intervene so early? And the answer to that is that actually she was accidentally and fortuitously diagnosed at birth. This young child, Ipek, was born in California, where she had a concerning newborn screen. Her newborn screening showed a low Treg count, which, as you know, is a warning sign for severe combined immunodeficiency, a genetic disorder which can be sometimes treated by stem cell transplant. Now, her low Treg count raised the concern for SCID, but in fact, a follow-up extensive testing by immunologist Dr. Jennifer Puck was actually negative for SCID, raising that this formally was a false positive in a way. However, further workup by neurologists Dr. Austri Foster-Barber and Dr. John Day at UCSF and Stanford showed that exome sequencing revealed the true cause. She actually had mutations in ATM, the gene which is the cause of Ataxia Telangiectasia. It turns out that AT is associated with a very mild immunodeficiency, not a severe combined immunodeficiency, but it was sufficient to cause this abnormal Treg count. And in this case, however, it left the family and her physicians in a bit of a quandary. This is a disease, AT, for which there are no treatments. In fact, the major symptom onset really was several years after this newborn period. And so this is actually not a type of condition which we are looking for in newborn screening. Formally speaking, this is something that we don't want because we are giving patients a diagnosis of something that's untreatable and before it even manifests. And there are potential psychological harms of doing that. However, in this particular case, one of her mutations came to our attention because this second point mutation, the C to T change, actually created a new splice site. And we know from the Armilosin experience what the importance of a new splice site is with respect to potential intervention. Creating a new splice site makes it potentially ASO amenable. So it was this fortuitous accidental diagnosis that led to her, us being able to recognize the opportunity to attempt an intervention for her. How extensible is this approach? I'd like to point out that the patient mutation that IPEC presented with is one that's not unique to her. It's actually prevalent as a recurrent mutation in the population. And a second AT child has now surfaced with that same mutation and is being enrolled in the investigational study of the drug that we've created. And additional patients are likely to surface. How many additional patients? Well, in fact, we have looked through whole genome sequencing catalogs of now over 300 cases of patients with Ataxia Telangiectasia. And we found that 10 to 20% of these individuals have mutations that appear to be amenable to this type of ASO interventional strategy. This work was done together with the AT Children's Project, Brad Margus, and the Global AT Family Data Platform. So I'd like to, through these examples, then end with some concluding thoughts to stimulate discussion. Really what this talk is about is the emergence of therapeutic platforms that are promising to transform the pace at which we can develop therapies for children with genetic diseases. Antisense oligonucleotides represent a growing family of molecules that can be customized for different purposes. And arguably approaches to doing the same thing, platform approaches to expanding and accelerating the deployment of gene therapies are beginning to gain traction. And other technologies that aren't here yet and immediately applicable in exactly this manner, but promise to someday, include CRISPR genome editing, with which we have the power to go in and correct mutations on an individual basis, no matter what the individual mutation is. And learning how to navigate these new possibilities is really the critical question. Charting a path to how to responsibly use these tools, one gene, one mutation at a time, is the name of the game for the next decade. And as we think about how these therapeutic possibilities will play out, we have to think about the impact of this on our general approaches towards identifying these patients, diagnosing them, and yes, screening. Our main takeaways from this are that, well, first, the disclaimer, Muscular and Atopexin are investigational drugs of unproven efficacy. However, successes like Nusinersen for SMA and these efforts to develop drugs like Muscular and Atopexin demonstrate their potential promise for use as therapeutic platforms for individualized medicine. And they really challenge us to confront the fact that genome sequencing can identify individuals who might be amenable to genetically targeted strategies that may not be existing at the moment that you sequence them, that perhaps you might be able to develop them just in time to have some option to offer, even if investigational in nature, for that individual. Timely identification here is critical if we are to serve our patients best. And we know that early intervention, especially in neurodegenerative conditions, but also importantly for neurodevelopmental conditions, is critical. And so therefore, thinking about what is considered an actionable newborn screening result in the light of these new possibilities and in our youngest patients, these are important conversations that we are at the position to begin preparing for. I look forward to those conversations with you. I want to thank the many folks who are involved in the work I've described in the Muscular proof of concept, manufacturing, toxicology, clinical trial. These were the key parties. I'd like to especially acknowledge Mila's Miracle Foundation, which supported much of this work. The work with Ataxia Telangiectasia also has involved a large team. I want to especially point out the support of the Baker family, as well as the AT Children's Project in moving that program forward. Thank you so much for your attention, and I look forward to the question and answer session.